So I'm going to focus more on biology, so that sets the stage for Sagar and Antonio to talk about things, and it might look like I'm more in favor of Antonio, but we'll see. So we have multiple genomic studies. You heard wonderful presentations early in the morning today, and what I wanted to highlight is that what we see may not end up being how it is used or applied. And just one example is that expression profile has been used to identify responses, and there's a 70 gene signature, 15 gene signature, 92 gene signatures, etc. So we've looked recently at whether these signatures can predict complete response, because if we can really predict complete response, we are able to pick right drugs for the right patients. And CR is the minimum endpoint for now, because everybody gets PR or better. So we need to look for CR. So we looked at 4 different studies. And good patient numbers, different methods and platforms, different treatments, but we're looking at CR, which was most of the time in these studies in 1/3 of the patient. And we used all sorts of bioinformatic methodology. And if you see the results, these are all different methods and all different endpoints. If you look at the accuracy, it's in the range of 60 to 65%. So if I flip a coin, it's 50%. If you do all this $1 million workup, it's 60%. Not good enough for us to pick the drugs. So what we need to know about all this data that you see is that there are significant limitations to what we can do with this. And so why would that be? Why are there limitations? And so, for example, when we look at expression profile, we are looking at RNA transcript and what it does to the cell at RNA level. What we are not including in the picture is what makes the cell behave the way it does. And the things that make myeloma cells, and for that matter any other normal or abnormal cell behave, are a lot of genomic correlates which drive the cells, from copy number changes to mutations to transcriptome modifiers, which you heard wonderful presentations in morning, and splicing. And we are not even touching protein yet. So we need to look at all this, which then may make us understand and predict some of these things better. What I'm going to show some of the data is about the gene mutational profile and how the clonal changes are observed and what is the heterogeneity in myeloma at one time point and over time. So we have studied 84 samples, exome sequencing, and in small number of patients, we have 2 samples evaluated. And standard algorithms are used, a lot of information on genomics and genetics, and most importantly, bioinformatics. And what we see is that we observe on an average 58 mutations per patient, and the frequency is variable from around 32 to 400 mutations per patient. If you look at the mutations in light of various cytogenetics and other changes, which are listed here for each of the patient, you could see there is no significant clear pattern. There is some increased occurrence of RAS mutations in people who have hyperdiploid myeloma, not statistically different. But other than that, it's all over. So there's nothing fixed. Number 2, if you look at the mutations, they're divided into genes previously implicated in myeloma, which are identified here, genes which are commonly implicated in other malignancy, and then there are genes here which are sort of new to malignancy and in myeloma, and those could be potential target. But nothing constantly seen here. Now, if we take an example of what we see in regards to mutation and how they occur, 3 common mutations, the commonest 3 mutations, which are in the range of 10 to 12%, KRAS, NRAS, and BRAF. And if we take individual patients, there are some patients where more than one of these genes from the same pathway are mutated. So we don't know what they're doing. Why do you need 2 mutations when one can do the purpose? And in some cases, like this patient, number 1 means all the cells have mutation, and then half of the cells and less. So in one patient, for example, here, most of the cells have both the mutations. Another patient where both of them are partial, and we don't know whether they're in the same cells or different. And then here, one mutation, the RAS mutation, is complete, meaning all the cells, and this one is in a minor clone. So they're all varied. And we need to understand what their purpose is, then to be able to target them. Now, I'm going to not show that can we target or not, and there is data that we can target, for example, BRAF mutations. But for the limited time, important thing to know is that even though anecdotal patient has been treated, in our analysis, we didn't find all BRAF mutation to be the classic V600E. There are many various other versions. And in some of the version, actually, there is a decreased BRAF activity. So the inhibitor may not function in these patients, although it will function and has been shown anecdotally that it works. The vemurafenib does work in V600E mutated myeloma. So more importantly, how these clonal changes are observed. Is there a single clone in myeloma, or there are multiple clones with different mutations? So this study provided a good data about the clonal spectrum or architecture in this disease. And how is it done is quite simple. So if you have one mutation in 100% of the cells, and then you have another mutation which is in 50% of the cells, you know off the bat that half of the cells have two mutations and half of the cells have one mutation. So there are two clones. And then if you have more mutations, this percentage changes. So there are more clones down the line. So for example, this patient has one mutation and one clone, predominantly looking, although this could be a separate clone. Here clearly there are two different clones, and here there are clearly three different clones. The major clone is not the clone which has all the cells with mutations. So you can see various spectrum. And in our analysis, using very stringent criteria, except one patient, every patient had more than two or more clones at the time of diagnosis. So even when we see the patient, there's already a lot of variation. And this is using extremely stringent criteria. If we move forward and try to see what happens over time in patients who had two samples, one at an early time point at diagnosis or early time point, second one subsequently between 4 to 6 months or beyond, what we observed is that in this one patient with all these different gene mutations, and red is mutation percentage in late sample, blue is early sample, there are clones which are showing mutation in early sample, but they disappear in the late sample. We don't see them again. And there are clones which appear in the new sample, but not in the early sample. And what we see in general is that there are significant dynamics between early sample to late sample. Some mutations go up, some go down, and others remain in between. So there's a really wide variation between how this changes. Now, we have further analyzed this to divide into 4 categories of their progression, how the evolution takes place. So to explain this figure, this is a subclonal fraction in early sample, meaning how many cells in early sample has each of these mutations. So one dot is one mutation. And this one y-axis is subclonal fraction in the late sample. So for example, this mutation is in 100% cells in the early sample, almost 100% cells in the late sample. Now, for this particular case, there is a cluster of clonal mutation in all cells and cluster of clonal mutation, subclonal mutation in some of the cells, but they are not changing. They remain more or less same. And so this particular patient did not change significantly between early and late. But the second version of how the change takes place is called differential clonal response, where cluster of clonal mutations are observed similarly in early and late, and they remain constant, and that's the trunk of the tree. But then there are subclonal mutations which are increased in the late sample. Initially, it is half of the sample. In the late sample, it has become 100%. And then there's subclonal mutation, which actually may have shrunk from 25% to close to 0% in the late sample. So if you draw the tree and the trunk, in the early sample, you have one branch which is thicker, the other branch is thinner. And in the late sample, the strength or the thickness of the branch changes, proportion changes. And how can this happen? It can happen because of number of reasons. It can happen because of the basic biology of the cell. A particular clone grows faster. Very possibly it can happen because of treatment, although some of our patients did not have treatment and still we saw this. In some of the cases, it's because of the microenvironment or epigenomic changes. So multiple things can lead to differential clonal response, which we need to study. Then the next type of progression we have seen in our patient population is linear evolution, where this mutation, of course, is seen in all patients, but then there is a subclonal mutation which is seen in a fraction of the sample, but more importantly, there is a group of cells or mutation which were not present in the early sample, but they have come up in the late samples with variable proportion. So what happens is that in the early sample, this is a trunk and a branch, but totally new branch comes up later on in a linear fashion. And there are molecular reasons how this can happen, and that's something we need to study. And finally, the most complex and probably most important that leads to lack of cure in this disease is branching evolution. So what happens is, this is of course the constant clonal mutation, but then there are a group of mutations which are clonal, which are present in the early sample, but they have disappeared in the late sample. And then there are a group of mutations which are not present in the early sample, and they have appeared in the late sample. So what happens is, in the early sample, there is one branch which disappears in the late branch, but a new branch, totally new branch comes up. And if you look at the frequency, and this is a small data yet, but if you look at frequency, we had around 5 patients who had no change, although this is using extremely stringent criteria. If we relax our algorithms, we do see changes in them too. But using this criteria, we see 5 patients here, and the rest are spread around here. And if we see, is there any connection with cytogenetics or other changes? Hyperdiploidy seems to have all the different changes, but 1114 translocation had most of the no changes. Does it any meaning to it, or just a small sample size? We have to wait and do larger studies, which is ongoing. More importantly, the 2 patients who had this more complex branching evolution both had extramedullary disease. Does it mean something? Again, a clue that we need to take and move forward. But this is the pattern of genomic evolution, and you can understand that as things evolve with different methods and different types, you might get resistance and more progressive, aggressive disease. Now, another way of looking at this clonal heterogeneity and evolution is we have performed IGH sequencing. So we have sequenced immunoglobulin gene. Mainly it was sequenced for MRD, but there is other uses for it. And as you will see, we have studied 297 patients. Some had RNA samples, some had DNA sample, and 5 patients have just DNA and RNA samples. What we observe is that there were 23% of the patient who had more than one IGH gene present in a given patient. So they were biclonal in 23% of the patients. Quarter of the patients had biclonal myeloma. The 77% had single clone. Again, using extremely stringent criteria of 5% or more occurrence of this clone. And this is the method how IGH was sequenced. Now, to give an example, what do we mean by this? Is that, for example, in this particular patient, had one clone with immunoglobulin heavy chain that was similar to the ancestral clone, where you could see that there are few mutations, which you see with hypermutation phenotype, and IGH had this phenotype. There's a second clone, which has not come from this clone, but it has come from this clone because it is the same mutation. So from an ancestral clone, 2 myeloma clones have evolved, but has some commonality to an ancestral clone. In another patient, there was totally different IGH. So in this patient, clone 1 is VH3, DH1, JH1. Clone 2 is VH1, DH2, and JH6. There's no common theme here. And that may suggest multiple things. We need to still dissect how this occurs. Does it mean that the second clone or 2 clones appeared at the pre-B cells, so in the earlier cells that has led to these changes, or something that more complex is happening molecularly? Interestingly, in this patient, there was an offshoot of a second clone. So compared to this one, there's another mutation here and a third clone, which was 1%. So there's a further increase in the clonal content with IGH changes. Now, interestingly, the 5 patients we have with both DNA and RNA had a very interesting finding. So 2 of the 5 patients had only one clone that we detected, and we take them out. But interestingly, the patients who had 2 clones, for example, sample 1, this patient had 1.5% content of one clone, but that 1.5% DNA was making 75% of the RNA. And the 97% was making the 25.5%. Similarly, this patient, 0.3% cells were making 16% RNA. And very interestingly, in this patient 5, 8% of the cells were making almost 100% of the RNA. So one other thing to remember is that if this patient's myeloma protein disappeared, we may have just affected this 8% cell. We don't have much idea about what happened to 84%, 85% of the cells. So this study does highlight that there are multiple clones and they change over time, and that we need to learn where do these clones occur, at what time point, the late occurrence, or its appearance at an earlier time point. So to conclude, we have described mutational spectrum. I didn't go through all the genes individually, but the spectrum is described, and except for MEK or pathway-related genes, none of the mutations are more than 10%. We unfortunately do not have Steve Trion's mid-88 in myeloma yet. What this means is that maybe we have subgroups of myeloma with very small occurrence of clones. There are SF3B1 myeloma, and there could be PYP2 myeloma, and so on. And what are the implications? There are many different patterns of evolution across patients, and point mutation and copy number, I didn't have time to show that data, they match. So sometimes the copy number may matter in what's happening. The important point is that we need to repeat genomic analysis. If we did the genomic analysis at the time of diagnosis, we may need to repeat it periodically to see what has changed and how we target them. And that may be an important point. And based on this, as we'll see a little bit tomorrow, we may have more targets and more pathways to go after for therapeutic purpose.